My name is Kayla Robinson. I will present on cadmium-activated NLRP3 inflammasomes mediate pyroptosis in raw macrophages. Cadmium is an environmental toxin that is deleterious to human health, affecting innate immunity. Cadmium toxicity in the liver, kidney, and lungs can be attributed to its long half-life. The NLRP3 inflammasome is a main sensory component of innate immunity, yet the mechanism of cadmium-induced immune dysregulation is not well known. Pyroptosis is a caspase-1 dependent pore formation inflammatory cell death. There are multiple triggers that can activate caspase-1. Gasdermin D and E also contribute to pyroptosis. Once caspase is activated, gasdermin D is cleaved by caspase-1 or 11, and gasdermin E is cleaved by caspase-3. We used macrophage cells and undertook this study to determine if cadmium induces the inflammatory form of cell death called pyroptosis in the raw cell line. The macrophage cells were treated for 24 hours with 5 to 15 micromolars of cadmium. The whole cell lysate was prepared and a Western blot analysis performed for pyroptotic-related molecules with beta-actin as the loading control, and confocal imaging was done using caspase-1 assay FLICA reagent. Macrophage cells were treated with cadmium nigiricin positive control or untreated. When FLICA is taken up by the cell, the caspase-1 is activated and cells will undergo pyroptosis. As indicated in figure 1 with nigiricin, all of the cells are shown brightly stained with caspase, so they are undergoing pyroptosis, and higher cadmium concentrations are undergoing pyroptosis, indicated by membrane blebbing and 10 and 15 micromolar cadmium treatments. KEAP1 expression from the endogenous NRF2 pathway is shown in the second figure. We observed the level of KEAP1 decrease with cadmium treatment and ROS produced, making it plausible to say that KEAP1 was degraded or ubiquinated. Without the antioxidant pathway activated in the control cells, the bands indicate KEAP1 presence. Figure 3 illustrates a reduced concentration in gasdermin D, though the band is very faint, but compared to the control, cadmium is decreasing the full length. The bands were present, yet we could not definitively conclude that cleaved gasdermin E plays a significant role in pyroptosis. Upon rerunning this experiment, a cleaved gasdermin antibody will be used so only the cleaved form is detected. To conclude, cadmium-induced ROS activates the NLRP3 inflammasome pathway, and pyroptosis was detected through caspase activity and cleavage of the gasdermins. We also plan to determine the mechanism of caspase-induced pyroptosis using a mouse genetic model. Thank you for listening.